Good day, everyone. Welcome to our next webinar in our ALS Canada webinar series for August. I hope everyone's having a great day. My name is Dr. David Taylor, the Vice President of Research for ALS Canada. And today we have a very special guest from the University of British Columbia. Jessica Morrice is a PhD student who received her Bachelor of Science in 2012 at Simon Fraser University in British Columbia, and has since been working as a student under the direction of Dr. Christopher Shaw, who's one of the preeminent scientists in the country looking at potential environmental factors and triggers for ALS. And so Jessica has been focusing on things like models of ALS in zebrafish, so hopefully we'll hear about that today. Certain cell types called microglia, which are supportive cells in the central nervous system around neurons and can ultimately affect the disease as it progresses. And as we've mentioned, certain environmental or toxic triggers. So without further ado, Jessica, welcome, and the floor is yours.
Thank you for the introduction, Dave. And thank you to ALS Canada for the invitation to talk about my research today. So the main focus of my lab's research is to investigate how environmental factors might be involved in sporadic ALS. I study how toxins can induce motor neuron degeneration using a zebrafish model. So today I'm just going to go over a brief background of sporadic ALS and different environmental factors involved in ALS, or that may be involved in ALS. I'm going to talk about how zebrafish can be a really great model of ALS, and then go over a couple results from a couple of the toxins that we use in the lab called bisphenol A or BPA and lead, and just describe the phenotype that we see in the zebrafish with respect to motor neuron degeneration, and then conclusions from our study so far. So as we know, ALS is a neurodegenerative disease which is characterized by the rapid progressive degeneration of motor neurons, which results in loss of motor function or paralysis. The majority of ALS research is focused on modeling different forms of familial ALS, and a lot of great work has identified a number of causal genes to familial ALS. However, 9 out of every 10 patients are diagnosed with what's called sporadic ALS, which lack evidence of a heritable genetic component, meaning that these individuals have no family history of ALS. So although sporadic and familial ALS patients are clinically similar, it's currently unclear whether they share a similar pathogenesis or disease pathway. So cases of sporadic ALS are assumed to be associated with different environmental factors. Epidemiological studies have linked risk factors such as heavy metals like lead, pesticides like organophosphates, industrial chemicals like aromatic hydrocarbons, and there's a bit of data to support endocrine disruptors. Also, athleticism has been implicated as a risk factor, and sort of one of the main examples of this is an Italian cohort of soccer players, or football players as it's called there, that developed a much higher incidence of ALS than what was seen in the rest of the population. Also, military service is considered a risk factor, head injury or head trauma, retroviruses, and consumption of cycad seeds has been linked to a form of ALS called ALS-PDC on the island of Guam. Cycad seeds look kind of like palm trees, although they're not, and they're a staple of the diet of the native Chamorro people. For a period of time around the 1960s, there was an extremely high incidence of ALS-PDC in the Chamorro people of Guam, and this is believed to be linked to the consumption of cycad seeds. Different groups have tried to identify the toxic constituent of cycad seeds, including our group. So epidemiological studies can only suggest associations or risk factors, and this doesn't necessarily mean causality. In order to further investigate the role of a risk factor in sporadic ALS, we have to ask what are the most effective ways that we can further investigate these. So models can be a really great way to investigate causality between potential risk factors and disease. Last month's webinar by Dr. Pichet Martel, and I apologize for my pronunciation, did a great job at discussing different models of ALS. Here, I'll just briefly talk about the common models of ALS, which are in vitro cell culture, C. elegans, Drosophila, mice, rodents, or rats, and Today I'm going to talk to you about why zebrafish are a really great model for this type of research. So zebrafish are particularly useful for modeling ALS because they have a similar and simplified nervous system as compared to humans. The genes implicated in neurodegenerative diseases are highly conserved in zebrafish. They have a large clutch size or number of offspring. They rapidly develop, which is really efficient for experiments. They're easily genetically manipulated, and they've been used extensively for toxin screening studies. Most importantly, and this is what I love most about zebrafish, is that they're visually transparent as embryos, which allows for high-resolution imaging of specific cells in vivo. This is an important feature of zebrafish, which is not possible to the same extent in higher vertebrates such as rodents. So this means that we can create zebrafish with fluorescent tags to specific types of cells, which we call transgenic zebrafish lines. Therefore, we can use different transgenic zebrafish to study what happens to specific cells as a time course analysis during disease. So below are images of transgenic zebrafish embryo under the bright field microscope to the left here and a fluorescent microscope to the right. The red arrow indicates the head region, and the trunk runs horizontally across each image. This specific transgenic line has a green fluorescent protein tagging motor neurons, and I'm going to talk pretty extensively about this line today. So as I said, zebrafish develop rapidly. The top left image is an embryo less than 1 hour post-fertilization, where the arrowhead is pointing to the embryo at the 2-cell stage. And within 24 hours, the embryos morphologically resemble a fish. So the majority of data I'm going to show you today is from 48 to 72 hours post-fertilization. My main research question is to determine if environmental factors such as toxins can induce motor neuron degeneration in zebrafish, and to investigate the mechanisms underlying pathogenesis. We currently house a number of transgenic or genetically modified fish. I'm going to give a brief introduction to each, but please feel free to email me with any specific questions. We frequently use the MNX1 line in our lab, which I showed you a couple slides ago, and this has a green fluorescent protein tagging a transcription factor or protein specific to motor neurons. So the motor axon in the cell body, or soma, fluoresce green in this line. We also have a line that's called the NBT line, and these have a blue fluorophore, or a blue fluorescent protein, tagging a protein specific to a type of programmed cell death called apoptosis. This system is controlled by neural beta tubulin, hence the name NBT, and in short, dead neurons fluoresce blue in this line. One of my favorite lines to use is the PU1 line, and these fish have a red fluorophore tagging a specific type of immune cell called a myeloid-derived cell. So in the central nervous system, this means that microglia will fluoresce red. We're also excited about the GFAP line in our lab, which has astrocytes tagged with a green fluorophore. This line is new to the lab, and we're currently growing this fish up to use for future experiments. So I will have results from this line next time. So using different transgenic lines, we can crossbreed to create double and triple transgenics, which allow us to study a number of different cells using distinct fluorophores or colors. So this is an example of a live imaging data that I collected from a double transgenic embryo by crossbreeding the MBT line with the PU1 line. So here, dead neurons are in blue and microglia are shown in red. Panels A through D represent a time-lapse image of a microglia, which is denoted by the yellow star in each image, approaching and beginning to engulf or phagocytose a dead neuron in the brain of a zebrafish embryo. The arrow in the last panel indicates an activated microglia after it has engulfed a dead neuron, and we know this because of the overlapping blue and red fluorophores, which appears white in the middle of the cell here. So a common question that I'm asked is, why do we model an adult onset neurodegenerative disease in a developing embryo? And the reason that we do this is based on the limitations of our transgenic lines, where we can only see these cells for a limited time, usually between 24 to 96 hours post-fertilization, which I will refer to in this presentation as the transparency time window. So heavy metals with endocrine or hormone disrupting properties are implicated in ALS and suggest a role for endocrine disruptors in disease etiology. At the beginning of my research, I performed a literature review to find toxins that can be dissolved in fish water and may cause motor defects in zebrafish. So BPA is an endocrine disruptor commonly used as an industrial plasticizer. Previous groups have shown that BPA leads to motor defects in zebrafish embryos, and we were interested to see if these BPA-induced defects were being caused by motor neuron degeneration. I want to emphasize and highlight that BPA has not previously been associated with ALS, nor are we trying to show that it is. The rationale behind using BPA as an environmental toxin is based on the premise that motor neurons may be vulnerable to toxic agents in a mechanistically similar manner, regardless of the actual toxin. So in order to test our hypothesis, we use the MNX1 transgenic zebrafish line that have motor neurons tagged with a green fluorescent marker. We first expose these embryos to a toxin dissolved in the fish water early in development. We then grow the embryo up until 48 hours post-fertilization, which is the most common stage to analyze motor neuron degeneration in zebrafish embryos. We then fix or preserve embryos in paraformaldehyde and perform imaging of the whole embryo, called whole mount imaging, using a specialized microscope called a confocal microscope, which allows us to image in depth in high resolution to produce a three-dimensional image. So next, we characterize the motor axon defects by measuring the length of the motor axon projection from the spinal cord and the number of axonal branching structures. These are zebrafish equivalents for what are similar to motor neuron degeneration that occurs in patients. So these images are of the zebrafish spinal cord, of a normal embryo at the top here, and an embryo undergoing motor neuron degeneration in this image. The spinal cord runs horizontally across each image, and the motor neurons project ventrally towards the top of the image. The yellow dotted line indicates how we measured motor axon length, and the red line indicates axonal branching. So if we find evidence of motor neuron degeneration, we then go on to analyze the neurodegenerative phenotype as a time point analysis. Briefly, we analyze the motor behavior, the integrity of the neuromuscular junction, which is just where the motor neuron meets the muscle and innervates the muscle, the microglia activation, and motor neuron cell death, which I'll talk about shortly. So this highlights the strength of a zebrafish as an efficient model to screen and clearly characterize motor neuron defects. So today I'm going to show you results from the toxins BPA and lead. So we first investigated if BPA has a dose-response effect. We used a range of doses and screened for mortality or embryonic death to find out what doses can be tolerated by embryos. We then tested if BPA has a dose-response effect on motor axon defects. So the axis on both of these graphs is referring to the treatment concentration. And the y-axis on the graph on the left is referring to the motor axon length. And the y-axis on the graph to the right is motor axon branching. So we can see that BPA has a dose-response effect on motor axon defects. Again, the reason that we care and the reason that we analyze motor axon length and branching are because these are markers of motor neuron degeneration in zebrafish embryos. We found that a dose of 50 micromolar BPA is optimal to cause reduced motor axon length and branching without causing substantial mortality, and the rest of the data I'll show you today is based on this exposure dose. So these are three-dimensional reconstructed images of motor axons projecting ventrally. So one set of motor axons projecting ventrally from half of the spinal cord and viewed from the cross-sectional plane. And this image over here is just to show the orientation of the view here. So up here we have the spinal cord in both images, and the yellow the yellow line is indicating the axon branch, or sorry, the motor axon. And we observed that BPA exposure causes reduced motor axon length and branching. Therefore, we've concluded that BPA exposure leads to motor neuron degeneration. So next, we performed a behavioral test called the touch-evoked escape response, which analyzes motor behavior. This is performed using a blunt object to give a tactile stimulation to the tail region. The natural response of the embryo is to swim away, and an abnormal or a reduced motor function, responsive motor function, is when the embryo does not move. So here I'm going to show, this is a control embryo. and we can see it with a normal motor response, and this is a BPA-exposed embryo. So we found that BPA-exposed embryos had an abnormal motor response or failure to swim at later stages of development, where 100% of embryos were unable to swim at 48 hours post-fertilization. And I will note that I screen all of my embryos for evidence of a heartbeat before I analyze them. So this embryo was alive, it just wasn't responding to the tactile stimulation. So the neuromuscular junction is where the motor axon innervates the muscle to cause movement. Here we can see the neuromuscular junction right here. And this is where the axon meets the muscle. The presynaptic terminal of the neuromuscular junction is located on the axon side of the neuromuscular junction, and the postsynaptic terminal is located on the muscle side of the NMJ. The integrity of the neuromuscular junction is important because a loss of integrity means that the motor neuron cannot innervate and transmit an impulse to the muscles, which results in a loss of motor function, and this feature is seen in patients. So to analyze the integrity of the neuromuscular junction, we fixed BPA-exposed embryos at 48 hours post-fertilization and labeled the presynaptic terminal on the axon side of the neuromuscular junction with an antibody called anti-synaptotagmin 2, which fluoresces green in these images, in these panels here. We stained the postsynaptic terminal at the muscle with a red fluorescent stain called alpha-bungaro toxin, and we can see that staining here. So here in these images, the spinal cord again is at the bottom of each image, and the axons are projecting ventrally towards the top of the image. The neuromuscular junction is located in this region. In the BPA group, or the BPA exposed group, we can see reduced staining at the postsynaptic terminal. So to analyze the integrity of the neuromuscular junction, we measure colocalization between these two fluorescent markers at the pre and the postsynaptic terminal, which appears yellow in the overlay of our control image here. We can see the BPA group has reduced colocalization between these markers, and when we quantified this, we found that BPA exposure causes significantly reduced neuromuscular junction integrity. So it's known that other cells play a direct role in causing motor neuron degeneration that's evident in ALS, such as glia cells. Glia are the neuronal support cells in the central nervous system, and glia such as microglia and astrocytes are known to have a critical role during ALS pathogenesis. So today I'm going to talk to you specifically about microglia, which are the resident immune cells of the central nervous system. So the morphology of microglia is indicative of their activation state. Resting or ramified microglia here present with long, thin processes or arms that project into their direct surroundings to sense stimuli. And once a stimulus, such as a pathogen or a degenerating neuron, has been encountered, the microglia will transition into a round amoeboid morphology, and this morphology is indicative of an activated microglia. So activated microglia have been shown to directly impact disease progression and have recently been shown to induce neurotoxic astrocytes. However, the specific role of microglia in ALS is still unclear. So understanding the role that glia have during early pathogenesis is crucial to further understanding the motor neuron degeneration that's happening and to developing targeted therapeutics. So this is a diagram of microglia activation based on their morphology, where the microglia are shown here in red. We're interested to investigate the activation state of microglia during early stages of neurodegeneration in our model. So here we've used a double transgenic line where motor neurons are tagged with a green fluorophore, and microglia are tagged with a red fluorophore. So this is a side or sagittal view of motor axons projecting ventrally from the spinal cord at the bottom of each image. We have found that microglia interact with degenerating motor neurons exist in a more reactive or activated state during early stages of disease or early stages of degeneration as compared to control groups. In this image of the spinal cord, dead neurons are labeled in blue and microglia are labeled in red. This is a time-lapse image showing that activated microglia engulf or phagocytose the degenerating neuronal soma at a later stage of degeneration. So this suggests that microglia perhaps sense pathogenic stimuli near the motor axons at early stages of degeneration, and we're currently investigating if microglia are directly involved in this degeneration. So this is an image taken under the confocal microscope of a live triple transgenic embryo with motor neurons labeled in green, microglia labeled in red, and dead neurons or neuronal debris labeled in blue. This embryo has been exposed to BPA, and the inset image here shows a microglia engulfing an axon terminal. We've reconstructed this image into a three-dimensional image. Here we go. And this allows us to get detailed information about the morphology or structure of cells and three-dimensional spatial localization of cells, which can be very valuable information for understanding how different cells are interacting. This is important because it allows us to distinguish between overlapping cells and cells that are actually interacting. So we then asked what time point motor neurons undergo cell death with respect to the motor axon abnormalities that we've characterized. To do this, we exposed the MNX1 line to BPA in fixed embryos at different time points. And then we stained for cell death using a non-cell death pathway specific marker called perpitium iodide. So here we can see the motor neuron soma in green and the dead cells in red. And co-localization between these two markers indicates dead motor neurons, which appear yellow in this panel. So we observed that BPA exposure results in increased cell death, where motor neurons begin to undergo cell death at 72 hours post-fertilization, which is after we observe the motor axon abnormalities at 48 hours post-fertilization. So speaking of cell death, there are a number of routes or pathways that can cause a cell to die, and knowing the pathway of cell death allows us to potentially target and block cell death. We're now focused on determining the pathway of cell death that's driving toxin-induced motor neuron degeneration. And our first inquiry into a specific cell death pathway has been on a type of programmed cell death called apoptosis, which can be thought of as kind of a type of cell suicide. So to investigate if apoptosis is driving cell death, we created a double transgenic line with motor neurons labeled in green, neuronal apoptosis labeled in blue. And after these embryos were exposed to BPA, we fixed them at different time points. Here, motor neurons soma are in green, and the apoptotic neurons are shown in kind of like a purple color. And co-localization between these two markers indicates motor neurons undergoing apoptosis. So here we can see similar levels of apoptotic neurons between the control and the treatment group. When we quantified this, we found that although apoptosis was slightly increased at the time point when cells undergo cell death at 72 hours post-fertilization, this did not reach significance. So this suggests that apoptosis is not the main cell death mechanism driving motor neuron degeneration in our, in our model. And we're currently investigating other cell death pathways. So when we put this data together, we can get a timeline of cellular dysfunction and dynamics during toxin-induced motor neuron degeneration. Whether motor neurons in ALS die in an anterograde or dying forward manner, or alternatively a retrograde manner where the cells die back from the muscle, is still an open question in the field. Using BPA as a model toxin, during early stages of pathogenesis, we have found that abnormalities at the motor axon and axon terminal that activated microglia interact with these abnormal motor axons. And during later stages, we found that motor neurons undergo cell death and that the microglia phagocytose the soma, or the cell bodies of the neurons. So our data seem to support that motor neurons degenerate in a retrograde manner, suggesting that the cellular dysfunction or pathology initiates at the motor axon or the neuromuscular junction, and this may cause the cell to die. And this has been shown by other groups using fall models, although we do need to further characterize this. So we've also found that apoptosis is not the main cell death pathway causing motor neuron degeneration, and in the lab, I'm currently performing experiments to look at the involvement of other cell death pathways. This data is important because if we know how the cell is dying, we can design specifically targeted therapeutics. So We've more recently started investigating the toxic effect of lead on motor neurons. Lead is a naturally occurring heavy metal, which has been used in construction, plumbing, batteries, leaded gasoline, among many other things. Lead has been shown to accumulate in the soft tissue and bones, and it's been previously shown to be toxic to motor neurons, and this has been known actually for quite a while. So evidence that suggests that lead is involved in ALS is based on evidence that shows high lead content in the blood, muscle, and cerebral spinal fluid of ALS patients. An epidemiological study showed that increased ALS incidence was distributed within the proximity of a lead smelting factory, and this happened in a gradient fashion. And I think this was in Missouri State that was performed. And then this is actually a Canadian effort of a meta-analysis of 9 case control studies that were investigating the role of lead in ALS, found evidence of a 5% increase of 5% of sporadic ALS cases may be attributed to lead exposure, and that lead exposure might cause around a 2 times increase of developing ALS. So again, there's much evidence to support the role of ALS as a risk factor, lead as a risk factor for ALS. So the data I will show you today are based on the methods that we have optimized using our BPA model, except we've exposed embryos to lead acetate. So to decide on a dose of BPA to use for modeling motor neuron degeneration around 48 hours post-fertilization, we performed a dose-response curve using different concentrations of lead. We first analyzed survival to determine the doses that would be tolerated for the duration exposures longer than 96 hours post-fertilization. We found that the highest concentrations were toxic within 24 hours post-fertilization, and I haven't shown those here. And that the lowest doses were well tolerated in our transparency time window. So we also analyzed the motor behavior using the touch-evoked escape response, as I described earlier. And we found that the lowest doses of 0.25 and 0.5 millimolar lead resulted in reduced motor function without causing substantial mortality within this transparency time window of our transgenics. So to investigate the effect of these exposure doses of lead have on motor neuron degeneration, we've analyzed the motor axon length at 48 hours post-fertilization. We found that exposure, and the y-axis here is the axon length. We found that exposure to 0.5 millimolar lead results in significantly reduced motor axon length. So we're now interested in determining the time point of motor neuron cell death from lead exposure using propydium iodide as a non-cell death pathway specific marker, as I explained earlier. So we have found that at 48 hours post-fertilization, there's no evidence of increased motor neuron cell death, and are currently investigating later time points. So far, these data are consistent with our BPA model. So in conclusion, we found that exposure to BPA or lead caused motor neuron degeneration in zebrafish, that exposure to BPA results in defects at the motor axon prior to cell death, that apoptosis is not the main cell death mechanism driving motor neuron degeneration in our BPA model, and that BPA and lead appear to be driving motor neuron degeneration in a mechanistically similar manner, although it's based on very preliminary evidence at this point. So together, this highlights zebrafish as a good model for screening toxins and investigating the pathogenesis underlying environmentally induced motor neuron degeneration. And we've sort of discussed this in more detail in a review that should be available soon online. So in the future, we're planning to further characterize the lead-induced motor neuron characteristics to determine the mechanism of toxin-induced cell death. And then I'm really looking forward to investigating the role of microglia, including how microglia are interacting with astrocytes during this degeneration. So this research may provide novel insights into the specific role of different cells during ALS pathogenesis. and may allow us to translate to novel therapeutic interventions. So before I go, I'd like to try and bring this full circle. Today I've talked a lot about toxins as risk factors for ALS. And while some cases of sporadic ALS may be solely attributed to environmental factors, it may be more likely that sporadic ALS is multifactorial in origin and involves a genetic predisposition or vulnerability where certain genes put one at risk for developing ALS. However, if these people may never develop symptoms or disease onset without sufficient exposure to an environmental stressor. So a common example that I think we can all relate to is that illustrates the sort of gene-environment interaction are individuals who are lactose intolerant. So people that suffer from lactose intolerance are genetically predisposed to have impaired lactose digestion and will present with symptoms if they're exposed to lactose. However, if these people are never exposed to dairy, they will never develop these symptoms. So the same may be true for sporadic ALS, and my lab is working to understand how environmental stressors may impact motor neurons to trigger degeneration. So thank you for your attention. Thank you to both labs, former and present members. A special thank you to Xiaohong Shan for all of her help teaching me how to use Zebrafish. And thank you to Dr. Francesca Perry and Dr. Pierre Drapeau for donating some of the transgenic lines that I showed you today. And I have time for questions, I guess.
Okay, thank you very much, Jessica. That was great. That was wonderful. And I think it's a question that a lot of people raise and think about all the time is what might they've been exposed to in life that might have caused them to get ALS, or whether it was something that they were susceptible to that someone else wasn't. Why did that happen? It's a question I think I get a lot as well from people, and they want to know why people aren't doing more of these types of studies. So I was wondering if maybe you could comment on how, like, what is the, or what your thoughts on the reasoning why there aren't a lot of people who are looking at environmental risk factors in ALS comparatively to those who are using genetic models?
I think that to sort of investigate environmental risk factors.
I think it's cutting out actually. Do you want to?
Oh, can you hear me? Can you hear me?
I'm not sure if I'm gonna find out if others can hear.
Uh-oh.
Can you? Oh, you can hear? Okay, I can't hear her answer. So maybe I will just go out to.
Is it working? Dave, I can't hear you. I don't know what's going on.
Okay, so I couldn't hear the answer, but I'm assuming it was wonderful.
I didn't even get to it. Sorry, because you said you couldn't hear.
Other people could hear your answer apparently, so it must just be my computer decided to go.
Oh, I mean, so, I didn't really answer because I didn't know that if anyone could hear me. But
okay,
I can attempt now.
Sure.
Okay, so I think it's an important point, and I think, I mean, the environment, there's sort of environmental studies are difficult because they're sort of reliant on, for a number of reasons. One of them is they're reliant on sort of recall bias and all these things, and there's sort of what's termed the exposome is all the environmental factors that we're exposed to during our life, and that's sort of infinite. So it's a very complicated venture to try and study environmental factors. But at least one way that our lab is trying to do it is basing these studies on on epidemiological evidence that's suggesting certain risk factors that we can go in and then further investigate. So it's just very complicated, and I think that's why more research isn't being done.
Yeah, I totally agree. I think that it's I think people are grateful that you are going after something like this, because it's definitely something people are interested in, but a very difficult thing to be able to approach and to be able to research. There are a couple other questions. One is, have you tried the cycad flower, the BPA that has been used, sorry, not BPA, BMAA, that has been used for mouse models of ALS PDC in the fish, and whether or not that had a similar type effect to the BPA in the lead?
So this is something that I'm excited to do, and we haven't done yet, is try the site. Well, we don't use the BMAA. We use a different constituent of the cycad called BSSG. So that's sort of our theory on what's the toxic component of the cycad. And we've used that in mice quite extensively, but not yet in fish. I'm excited to do it. It's just sort of, we don't really have the funding for that right now, but definitely is a future interest.
Okay. And then one last question is, this one's from me, is I think it's really interesting that you're starting to look at how you can merge genetics with the environmental toxins, and how the environmental toxins might become triggers, or something that may not be a trigger unless someone has the right particular key cut of their genetics. And I guess the question is two things. One is, what led you to lead? Wow, that's a weird way of putting it. And the second one is, do you think there's ever a way in which zebrafish could be used to potentially screen for environmental factors if you were able to discover some sort of genetic enhancer elements or something like that that can be altered by an environmental toxin to trigger that genetic pathway to make someone susceptible? I don't know if that was very clear.
So to get to your first question, why lead? So when I'm sort of screening for, not screening, when I'm looking for environmental toxins to screen in my embryos, I perform a literature review. So because we have to administer the toxins in the fish water, it has to be water soluble, or I basically have to be able to administer it to the fish, to the embryos. So something that's easily kind of delivered to the embryo. And then also I look for previous studies that have shown toxic factors that have induced sort of toxic neurotoxicity for motor defects in zebrafish. So base it on, I guess, previous groups' work. And then I try and also, if I usually have a pretty big list at that point. I try and find toxins that are implicated in ALS. So there's actually a lot of evidence for lead in ALS, and we're pretty excited about studying this one. So that's how I ended up on lead. And your second question was a bit longer, and I can't remember it.
It was essentially that you had started to talk at the end about how you wanted to look at how environmental factors might become triggers for people who are specifically genetically susceptible for the disease. And that's, I think, the coolest avenue of untapped research in terms of something that we as a field have not really approached yet or found a good way to do. And I'm wondering if the question is, if you were able to find through all of the work that's being done with whole genome sequencing and all of the data that's now becoming available, if you're able to find ways in which you might be able to someday screen for a series of chemicals or toxins that could be environmental factors that would then match up with the genetics for certain individuals, kind of instead of personalized medicine, it's like personalized toxicity. So can you foresee a time when maybe, the first step is we understand the genetics of the disease, and then the second step is, can we actually use that to screen for who is susceptible for what particular environmental toxins?
Right, yeah, that's you're kind of going along the lines of something that we're really excited about in the lab, and we've just started. And I probably can't say too much, but we formed an exciting collaboration with a group to kind of pursue the gene toxin aspects of ALS, and we're sort of planning on modeling these different risk genes interacting with different toxins in our zebrafish because it's such a good system for it. So that's something, yeah, that's sort of where the project is headed right now, but I don't know how much I can say.
Yeah, no worries. I'm just trying to highlight that I think that there's a lot of potential, for the audience, that there's a lot of potential in this to be expanded into something that can be quite huge in terms of the concepts that it can approach. But,
right,
thank you
yeah.
very much. And, I think that's all the questions we have from the audience. So we really appreciate your time today and for your presentation. And thank you to everyone who joined us online. And we look forward to seeing all of you again in September for that webinar. So thanks, Jessica.
Thank you so much. Thank you.
Okay, take care, everyone.